thank you so much for this opportunity of speaking in this forum. We are local, so when we knew about this meeting, we jumped to the opportunity to share our research interests and our finding in this forum. So I am a clinical genetics trainee. I don't have a PhD, and right now I'm doing a clinical research year under the mentorship of Miao He. And I'm going to share with you today very interesting patients, and I hope to spark your attention and your interest, even if everybody's hungry and thinking about lunch. So I promise to be brief, and I will not go over 20 minutes. So the congenital disorders of glycosylation are a rapidly growing disease family. There's approximately 100 disorders that have been identified. And it is estimated that about 1 to 2% of the human genome, it's a glycosylation-related genes. So there's a lot of genes there, a lot of opportunities where things can go wrong and human disease can happen. It was reported that even in 2013, like every 17 days, a new glycosylation disorder was found. And this is due to the new advances in next generation sequencing. So all of the sequencing data of patients' exomes and genome have elucidated all these disorders. So most CDGs are due to hypoglycosylation, and the classic defects are on the N and O glycosylation pathways, or both pathways. But there are also disorders that affect the synthesis of lipid-based pathways, and this is the area where we're going to talk about today. So the GPI, or glycosylphosphatidylinosytol anchor biogenesis defects, are a subtype of the CDGs, and they involve the intersection of two pathways, which are the lipids and the carbohydrates. So the GPI anchor was a structure that was discovered about 20 years ago in protozoans, actually. And it has a glycan core. It has a phosphoethanolamine linker and a phospholipid tail, and it's a very interesting post-translational modification that is highly conserved among eukaryotes. And these GPI anchor proteins perform a diverse set of functions, including cell signal transduction, cell adhesion, antigen presentation. And these are just some examples of GPI anchor proteins. There are over 200 proteins that require this post-translational modification. So, for example, CD59, which is a surface marker and is involved in complement regulation. There is alkaline phosphatase, which is an enzyme that is present in the liver and in the bone, and there are many cell surface hydrolases as well as many neural cell adhesion molecules. So as you see the cases that I'm going to present, it's going to be making more sense why these patients have clinical manifestations in basically all systems. So very briefly, the GPI biogenesis pathway has basically 3 stages. The first stage, the whole synthesis occurs in the endoplasmic reticulum. The first stage includes about 10 proteins, and these synthesize the precursors of the GPI anchor. Then this whole structure is flipped into the luminal side, and various sugars are added. And in this particular step here, which is the step that we're interested in, phosphoethanolamine is added. So there are about 12 genes, and they're all here in red, in which human disease have been reported, and these have various phenotypes. Most of them have congenital anomalies. They have very specific neurological phenotype, including epilepsy, developmental delay, intellectual disabilities. So this is what we're interested in. PIGN encodes for the GPI ethanolamine phosphate transferred 1, and it transferred this phosphoethanolamine into the structure. The first family with this disorder was actually described in 2011. This is a very rare condition. Not a lot of reports are out there, and it was found on an Israeli family in which there were several children affected with this condition, and they all manifested with developmental delay. They had dysmorphic features, and they have multiple congenital anomalies in different body systems. And they mapped the gene into this locus 18q, and they found the disease-causing mutation in the PIGN gene. The second report was a report in 2014 of two Japanese siblings who also had a similar neurological phenotype with the seizures that were very hard to control. They had some dysmorphic feature, but they also had a new finding of progressive cerebellar atrophy. And the disease-causing mutation was also found in the same gene. The third report was an unborn fetus that had several congenital anomalies, including diaphragmatic hernia, which is like a hole in the muscle that divides the thorax and the abdomen. And they also found a mutation in PIGN gene by exome sequencing. So this is just some pictures of the fetus, and you can see the congenital malformations in basically all systems. And this is a latest report that came up like 2 months ago, and it's a patient from North Carolina of Hispanic ancestry that also had very low muscle tone, some genital abnormalities, visual impairment, and some abnormalities of the chest. And now we're going to move on to our cases. So we had 3 patients at the Children's Hospital of Philadelphia. This is a little girl that I saw during my first year of training. She had a history of seizures since the first day of life that were basically intractable. So she spent her whole life in a hospital. She never went home. She had very low muscle tone, and she had multiple birth defects. She also had what we as geneticists call dysmorphic features, which means that the facial features are a little bit different if you compare to the child that is not affected. So she had the eyes, you can see that are a little bit sticking out. The distance between the eyes was a little bit wider than normal. The helixes of the ears were a little bit thick. The upper lip was a little bit tented and thin, and the chin was small. She also had some of her cranial sutures that fused before time, and that's what we call craniosynostosis. One finding that particularly struck us, because as geneticists, we often see patients with multiple clinical manifestation in all systems. So when you have a child like that, the finding that is least common is sometimes what clues us in. And this particular patient had fingers that were very short, and the last phalange was extremely small. And we did x-rays, and we can also see that the phalanges are tiny. And it was actually absent on the 5th digit, and the same was present on the feet as well. And unfortunately, this patient at the time passed away without a diagnosis. But we did send whole exome sequencing to a clinical lab, and that was pending. And after, as I said before, whole exome sequencing, which is basically a technique in which you can sequence the coding parts of the genes instead of the whole genome, it's a very good thing, because before that we had to think about the gene that we wanted to sequence, so we basically went looking for that. But whole exome sequencing allow us to sequence all exomes. So we did find an answer for this patient. And we found 2 mutations in this gene, one affecting a splice site and another missense. And after we diagnosed this patient, a group of genetics colleagues remembered this patient that they saw about 6 years prior, even before I was a fellow at CHOP. And this patient also passed away without a diagnosis, but fortunately we had some fibroblast cells from a skin biopsy, and she presented in a very similar fashion. She also had very low muscle tone, she had seizures and multiple congenital abnormalities and some differences in the way that she looked. And she had the same finding of the hypoplastic terminal phalanges. She has some cardiac malformation as well as imperforate anus and very particular uterine malformations called didelphys, in which the uterus is basically divided into 2. And for her, in her case, we went straight to the PIGN gene, and we find a mutation that was previously described, the first one, and the second one that is a novel mutation. And the third patient is a patient that had a milder phenotype. This patient is still alive. The prior 2 patients passed away. They were severely affected. And she does have some facial appearance differences. She had very low muscle tone, and she also had very difficult to manage epilepsy. These are just the description of her facial findings, and she also had mutations in the same gene. So what are we interested in? So in our lab, we are interested, and I am particularly interested in the clinical characterization or the definition of the phenotype of this condition. We are looking into biomarkers of disease, and we want to learn more about the inside of the pathogenesis of these conditions, and hopefully this will allow us to think about potential therapists. So what have we done so far? So, before that, before the advances in the next generation sequencing, the only condition that was known in this the GPI biogenesis pathway was what we call PNH, of paroxysmal nocturnal hemoglobinuria, which is due to a somatic mutation in PIGN, which is the gene that encodes for one of the first steps of the GPI anchor biogenesis. And basically PNH, what happens is the GPI anchor is not properly synthesized, so there are some cell surface proteins that are important for the integrity of the red blood cells. So these patients basically have hemolysis. And the way that PNH has been diagnosed classically is by flow cytometry analysis of these surface proteins. So we thought about doing this as a biomarker, and we searched the literature, and we found some reports of patients with defects in these pathways that had flow cytometry in different proteins done. Most of them were done in either in blood cells, but for our patients, we had fibroblasts. So we set to develop a flow cytometry or fluorescent activated cell sorting protocol to prove GPI anchor deficiency in these patients. And we also are interested in examining the functional impairment of compound heterozygous mutations by analyzing the surface expressions of various of these proteins. So we chose these three proteins, which are proteins that we know are expressed in the fibroblasts, and they have different functions. We purchased fluorescent antibodies, and we did flow cytometry on the two patients that we have cells available, and we can clearly see a shift when we compare it to normal human fibroblasts in the expression of CD59 here, and we also have conclusive data for the expression of CD90. We also did flow cytometry in other proteins like CD16, but we didn't see very clear shift. We also tested fluorescent activated proallorizing, but it didn't work very well either. And in conclusion, we do believe that patients with this mutation can present with variable clinical phenotypes that affect different body systems. And the consistent features in all the cases that we have in our hospital and the previously reported cases are mainly the muscle tone, the hypotonia, the seizures, the developmental delay, facial dysmorphism, congenital abnormalities may or may not be present. We do believe that FACS analysis in affected patients can help prove the deficiency of the GPI anchor biosynthesis. And right now we are working on functional studies at our lab at CHOP to characterize the consequences of each one of these mutations. And I want to thank all my lab members who are all here in the audience. Shoeli, who is speaking a little bit later this afternoon and taught me everything from cell culture, because before this I had no basic science experience. Rayhan, who is always there helping us, and Yolanda Foster, who is actually working on molecular cloning and overexpression systems to characterize each of the mutations that I presented, and my mentor Miao He for taking this fellow with an idea and no experience, and Awo Akosua Lehman, who works in the immunology research group, and she taught me everything I know about flow cytometry. So thank you very much.
Great job, you stayed on time. Quick question or two? Yeah, good. All right.
We did work on patients that had sialidosis and galactosidosis, where we looked at fibroblasts. And even though they do express quite a bit of sialidations on the receptors, if you and these sialidoses have a defect in the U1 sialidase activity. This particular study here with the GPI, even though it's involved in multiple different receptor signaling, you seem to target the immune system here. Do you think this particular area is more immune deficiency?
No, the reason why we decided to do it is because it was a technique that was easy to do and where proteins that we knew required a GPI anchored, and it was a way that we thought we could prove in a very easy and effective way that in fact these patients had a GPI anchored deficiency.
Yeah, I'm just thinking here in terms of possible treatment, if any. And if you understand the mechanism of how it works, then maybe you can do a target. In particular, when we looked at, for example, the sialidoses, they do express quite a bit of sialic acid. And if you can activate, for example, NU4 sialidase, you can cleave these sialic acids and get a normal phenotype. And I'm just wondering if there's some possibility with regard to this.
It's a good idea. It's an interesting thought. We haven't thought about that aspect, so I'm glad that you bring it up. And if anybody else has any ideas, you can find me during the breaks or after lunch. And I believe my email address is on the abstract book. So I'm happy to hear any suggestions.
I think some of the people are going to come and have beer with me over my talk. Maybe you can join me.
Okay, thank you.